Welcome to the lecture series Cell Communication. I'm Janine Post of the Developmental Bioengineering Group of the University of Twente. Here I will discuss ion channel coupled receptors. Illustrations in this video are based on the book Essential Cell Biology, unless otherwise indicated. The information given in this lecture can be found in chapters 12 and 16. In the series Cell Communication, I give 5 short lectures. This lecture is the first of 3 separate lectures on the most common receptor types, ion channel coupled receptors. The basic principle of signal transduction is that an extracellular molecule binds to a specific receptor, thereby activating intracellular proteins that influence many cellular processes, including cell differentiation, migration, proliferation, etc. Ion channel coupled receptors, or ligand-gated ion channels, are a combination of a specific ligand binding site and an ion channel. Upon ligand binding, the ion channel will open, which allows transport of specific ions across the membrane. To understand the need for the ion channels, we need to backtrack to chapter 12 of Essential Cell Biology. We know that lipid bilayers, such as the cell membrane, are impermeable to ions in most uncharged polar molecules. The ion concentrations inside a cell are very different from those outside the cell. Difference in the concentration of inorganic ions across the cell membrane create a membrane potential. Cells contain 2 classes of membrane transport proteins, transporters and channels. Solutes cross membranes by either passive or active transport. Passive transport, and thus the direction of the transport, is influenced by both the concentration gradient and membrane potential. Water moves across cell membranes by a process called osmosis. The hydrophobic interior of the lipid bilayer creates a barrier to the passage of most hydrophilic molecules, including all ions. Diffusion of molecules is slow and uncontrolled, so for the import of most molecules, cells are dependent on active transport. As shown in the figure, there is a decrease in diffusion based on the properties of the solutes that are shown from top to bottom. Ions and other small but uncharged molecules cannot diffuse and need to be actively transported. There are different types of cell and organelle membranes in terms of molecular composition. Each type of membrane has its own characteristic set of transport proteins. This determines exactly which solutes can pass into cell or organelle. Inorganic ions and small polar organic molecules can cross a membrane through either a transporter or a channel. A channel, shown here, forms a pore across the bilayer through which specific inorganic ions, or in some cases polar organic molecules, can diffuse. Ion channels can exist in either an open or a closed conformation, and they can transport only in the open conformation, as shown here. Channel opening and closing is usually controlled by an external stimulus, or by conditions within the cell. A transporter undergoes a series of conformational changes to transfer small solutes across the lipid bilayer. Transporters are very selective for the solutes that they bind, and they transfer them at a much slower rate than do channels. Different types of gated ion channels respond to different types of stimuli. In the top panel we see the closed, and in the bottom the open conformation of the channels. Depending on the type of channel, the probability of gate opening is controlled by a change in the voltage difference across the membrane, binding of a ligand to the extracellular phase, binding to the intracellular phase of a channel, or by mechanical stress. Chapter 12 discusses examples in which each of these channels is used, like in hearing and in different types of nerve cell signaling. So for more information, I refer you to read chapter 12. Ion channel coupled receptors, or ligand-gated ion channels, are a combination of a specific ligand binding site and an ion channel. Upon ligand binding, the ion channel will open, which allows transport of specific ions. Ion channel coupled receptors change the permeability of the plasma membrane to selected ions and thereby alter the membrane potential. And if the conditions are right, they produce an electrical current. These receptors are responsible for the rapid transmission of signals across the synapses in the nervous system. When a neurotransmitter in the synapse binds an ion-coupled receptor, the receptor alters its conformation, thereby opening a channel, allowing for transport of specific types of ions, like chlorine, sodium, potassium, calcium, or combinations of ions. Depending on the electrochemical gradient, the ions rush in or out of the cell, creating a change in the membrane potential in milliseconds. Many animal and plant toxins interfere with the function of ion channels. Although our understanding of these toxins and how they work remains incomplete, physiologists and pharmacologists have exploited them to explore the function of ion channels. An example of this is charybdotoxin, a scorpion toxin, named after Charybdis, the daughter of Poseidon. Charybdotoxin is a peptide that specifically blocks a calcium-activated potassium channel. This results in intense pain, numbness and tingling, tachycardia, high blood pressure, and involuntary or unusual head, neck, and eye movements. Because of the binding of charybdotoxin to its channel, its discoverer, Chris Miller, was able to deduce the shape of the calcium-activated potassium channel. This was confirmed much later by X-ray crystallography. In 2013, the complex of the potassium channel with the toxin was imaged by Banerjee et al. You can see that in the schematics below. More information on the imaging or the function of these toxins on channels can be found in the references indicated. Thank you for watching this video. For questions, please respond below or email me. And tune in for the next video on G protein coupled receptors.